Right. Good afternoon to all the dignitaries. Today's guest speaker, Professor Christopher Sir, and Principal, Government Degree College for Women, Jagityan, Satnaran Sir, and all the participants of today's webinar, that is Emerging Trends in Green, Red, and Yellow Biotechnologies by Professor T. Christopher Reuben. Sir Sir is a Dean from Chaitanya Dean to be University, Hanmakonda. Now I request Principal, Government Degree College for Women, Satnaran Sir, to address the gathering. Over to you, sir.
Thank you, madam. Good afternoon, everybody.
Good afternoon, sir. Good afternoon.
Good afternoon, sir. Today, we are conducting emerging trends in green, yellow, red biotechnologies with association RBVRR College, Hyderabad, and Principal Madam Achyuta Devi Garu, and HOD Rajini Devi Garu, and resource person Christopher Garu, and my dear colleague Tirupati Garu. It is a good opportunity to participate in this webinar, sir. Today, we know about the green technologies and biotechnologies, yellow and red technologies, and given message by the Christopher Gar. Thank you, sir.
Thank you, sir. Thank you.
Okay, sir. Thank you very much, sir.
Thank you, madam.
Now I invite our respected Head Department of Botany, RBVRR Women's College, to speak few words about today's webinar.
Good afternoon to all respected members of management, both college principals, faculty, and my dear students. I welcome you all for today's webinar on emerging trends in green, yellow, and red biotechnology. I welcome Professor Christopher Sir, Dean, Faculty of Science, Chaitanya deemed to be University. Thank you, sir.
Thank you, madam. Thank you.
Thank you very much, ma'am. It's our immense pleasure to invite sir. And we are very happy, sir, to accept our invitation and to be present here as a resource person. Now I invite Dr. P. Manjula, Assistant Professor, Department of Botany, to introduce today's resource person, Professor Christopher Sir. Over to you, Manjula.
Thank you, madam. Good afternoon, one and all. It gives me an immense pleasure to introduce today's guest speaker, Professor Christopher Reuben, sir, Dean, Chaitanya deemed to be University. Sir completed MSc and PhD from Kakatiya University and awarded University Research Fellowship and Senior Research Fellowship from Kakatiya University and Research Associateship from University of Delhi. Sir held different positions during tenure of his working, like course coordinator of medical transcription during 2008, additional controller of examination during 2013 to 15, head of the department during 2018, director foreign relations officer during 2017 to 18. Presently, sir is dean of research development, Chaitanya deemed to be University Warangal. Sir has 15 years of teaching experience and 20 years of research experience in the areas of plant tissue culture, genetic transformation, molecular markers, and SAR has published 48 national and international journals, and SAR has to his credit 3 major research projects. Currently 8 PhD scholars are working under SAR's supervision. SAR attended 28 national and international conferences, and SAR has delivered 25 invited lectures in various colleges. Through this brief introduction, now I hand over the session to sir. Thank you, sir.
Thank you, Manjula. Sir, before sir's talk, I welcome our respected secretary cum correspondent, Professor Sudarshan Reddy, sir. Sir, hello, Sudarshan Reddy, sir. Nas?
Yeah, ma'am. Sir can unmute, ma'am.
Yeah, okay. Sir, you can unmute yourself, sir. You can speak few words on this occasion. Sir, Sudarshan, sir.
Yeah, good afternoon. Am I audible?
Yes, sir, you're audible, sir.
Am I audible? Yes.
Yes, sir.
Let me at the outset apologize for the delay in joining in the webinar, because I was away traveling from Madhapur and just reached. And I am very happy that this collab. Firstly, I thank Dr. Satyanarayana Garu, Principal of Government Degree College for Women, Jagityal, because this collaborative effort was the initiative of the previous principal and our own principal, and because of the efforts of the departments of botany of R.B. Vera Women's College and the Government Degree College, Jagityal. I must mention the name of Dr. Padala Tirupati, who made a lot of effort in establishing this togetherness between the two institutions. This is wonderful. And this has been, in fact, this was the result of the pandemic situation. This was, in fact, started in the beginnings of pandemic situation, and which is continuing. And this is a great effort. And apart from this, the two institutions also made effort to provide free coaching to the students of both the institutions and also others, as far as PG coaching of CPJET is concerned. I am told a good number of students have been selected in the just concluded what you call the admission process. So I am on behalf of the management, I also welcome the guest speaker of today's webinar, Professor Christopher Reuben, formerly Kakatiya University. I am just now told he is now with Chaitanya Deemed University of Warangal, and he is going to speak on wonderful areas like emerging trends in green, red, and yellow biotechnologies. I hope the students of both the institutions and others will make use of this lecture and maybe fruitfully benefited. And such an effort will continue in the days to come, in fact, in the years to come. I appreciate the efforts of the heads of both the colleges, Rajani and Padala Tirupati, and also the principals and other faculties who are involved in this kind of academic effort. And I once again, on behalf of the management and on my own behalf, welcome the guest speaker as well as the principal of the Government Degree College for Women, Jagityal. Thank you very much for giving me this opportunity to share some of my ideas with you. Thank you very much.
Thank you very much, sir, for your wonderful ideas and suggestions for these webinars. Now, I invite today's guest speaker, Professor Christopher, sir, to hand over the session, to take over the session, sir. To you, sir.
Right. I can share the screen, madam. Enable me to share this.
Okay, sir. Nazneen, please enable sir to share the screen.
I have already done that.
Yes, sir. Sir, you can share, sir.
Most disabled participant screening share. I don't know. Madam.
Nazrin, sir is getting host as a disabled. Enable.
Yes, ma'am, I'm checking, ma'am. 1 minute.
Okay.
Sir, please try now.
Yeah, yes, madam. Thank you. Thank you. Thank you so much. Yeah. Yeah. Thank you, respected Secretary Sudarshan Reddy garu, Principal of RBPRR Women's College, Narayanguda, Hyderabad, and also Principal of Women's Government Degree College, Women, Jagityal, for giving me an opportunity through Rajini Madam and through Kavita Madam and also Tirupati for this opportunity to speak to UG students. So the topic that I have selected is not that new, but I will try to present what is green biotechnology and what is yellow biotechnology and red biotechnology. I'll be trying to present the information during this course of this discussion. And then one more important thing is, I am very grateful to all of you for giving me this opportunity for presenting this particular study. Okay, so my acknowledgements are to Dr. G. Sudarshan Reddy, is the Secretary, Dr. J. Achita Devi, Principal of RBPRR Women's College, Hyderabad, Dr. Y. Satyanarayan Sir, Principal, Government Degree College for Women, Jagityal. Special thanks to Dr. Rajini Reddy Madam from Department of Botany, RBPRR Women's College, Hyderabad, Dr. Kavita, Dr. P. Tirupati, and Dr. P. Manjula. So I am grateful to every one of you for giving me this opportunity. Right. So I am going to show you some very important things pertaining to food, pertaining to medicine. Father of genetics is Mendel, 1865. He laid the foundation for plant breeding. He was responsible for developing the system of monohybrid ratio and dihybrid ratio. So he was one of those persons who has contributed to the food production in the world. The next we come across is Alexander Fleming. Alexander Fleming, 1921, was responsible for the discovery of antibiotics. First antibiotic was penicillin. Up till that time, there was no antibiotic. It was used, it was not known to them, but it was a great effort made by Alexander Fleming, who has finally isolated antibiotic, which is the suppressor of bacterial infection and other infections caused by bacteria. And Norman Borlaug is father of Green Revolution. With him, he worked on wheat. He's from Mexico. With him, he was awarded Nobel Prize for his contribution to food production. So these three persons have a lot of things to play for food and medicine. So in further lecture also, I will be trying to highlight some of these developments that have originated from these persons. Okay, and then from our country, it is B.P. Pal. He was the first Director General of Indian Council of Agriculture Research, and also is the first plant breeder and agronomist. So he hails from Delhi, and then he has contributed a lot to food production. Then we also have M.S. Swaminathan, who is the father of Indian Green Revolution. India was deficit in food production, but after MS Swaminathan, in association with Norman Borlaug, were able to make India from a deficit to a surplus country. Thanks to these people who have contributed much for the food production. Today, we have sufficient food in our country. It is because of Swaminathan's contribution. The person in the center is Sir Ronald Ross. He was also in India. He received Nobel Prize in Physiology of Medicine in 1902 because he first discovered that malaria enters into human system through mosquitoes. So that was the reason I have given all the three names, D.P. Pals, Ronald Ross, and MS Swaminathan. And the other great scientists of our country, I call them as biotechnologists or plant scientists. One is Jagdish Chandra Bose. Jagdish Chandra Bose is onto this side, onto the right side. He was a person who actually recognized plant movement, and he developed an instrument called chrysograph. Then we have Panchanan Maheshwari, who was a great embryologist of our country. Then we have Sipra Guha Mukherjee. She was the first lady scientist from our country who actually studied somatic embryogenesis in Datura, in anther culture. That's the first contribution in tissue culture. Professor Ashish Datta was also a professor of JNU and also vice chancellor of JNU. And then he isolated the genes for protein from crop plants, and then he introduced them into other crops. Then Maskar Haas from National Chemical Laboratory, Pune, he worked on in vitro flowering in bamboo. Bamboo flowering happens once in lifetime. But this group in NCL Pune was able to achieve flowering inside a test tube, inside a culture tube. And that's real achievement. Then Deepak Paintal, who was the vice chancellor of Delhi University, he developed male sterility by using genetic engineering method. Then Anand Kumar, who was responsible for developing BT brinjal, which is not released in our country yet. Then Professor A.R. Mehta is from Baroda, Gujarat. He was responsible for developing secondary metabolites from tissue cultures. Professor Lakshmi Sita from IIS, Indian Institute of Science, Bangalore, she was involved in micropropagation of sandalwood. Sandalwood is endemic to our country. A very, very rich vegetation we have in our country. Here you see a photograph. She's interacting with Prince Phil in IISc Bangalore. Then Professor G.M. Reddy is also one of the well-known plant tissue culturists of South India. He is from Osmania University. So I have grouped some of them. So they have made rich contribution to growth of the plants. Okay, and then father of biotechnology is Louis Pasteur. We also know that Louis Pasteur is also father of microbiology. So it has its roots in biotechnology. Then the word biotechnology was coined by Erike, Karoli Erike. He's a chemical scientist, a chemist. So these two people have made the beginning of this. Okay, and I also have some important names here. Herbert Boyer and Cohen. These were the two people who actually developed the rDNA technology for crops. Charles Arnitzen was responsible for producing tomato, which has a long shelf life, flavor sour tomato, you call it. You can store it for 3 weeks outside a refrigerator. And then Ingo Potraikas was a scientist who developed golden rice, where rice will have the gene for provitamin A. That is how Ingo Potraikas from Switzerland has been able to develop. And then Phil Leder and Tim Stewart were responsible for developing a mouse model for cancer. So that is how they have contributed. These two people that we see here, Carey Mullis, a great contribution to science, to experimental science. He has developed polymerase chain reaction in a thermocycler. He has developed, he's invented thermocycler. Today we are using RT-PCR for detecting coronavirus. So PCR was actually developed by Carey Mullis. And among all important contribution was Margaret O'Day Hoff. She is the mother of bioinformatics. Very important contribution. She has developed an atlas for proteins. So she's the mother of bioinformatics. So students can remember this issue when you go for writing an exam. Okay. Right. So biotechnology, all of us will come across this word biotechnology. But in the word biotechnology, there are 10 classes of biotechnology, 10 areas of biotechnology. This was done by UNESCO. They've given colors, so you call it as 10 colors of biotechnology. First is the green biotechnology pertains to agriculture, where you have crop improvement via breeding program, biotechnology, CRISPR-Cas technology, all those things. Then you have yellow biotechnology, where we have food and nutrition, fermentation, biotransformation, and insect biotechnology. I will be today dealing with insect biotechnology, not the first two. A lot of information is available with fermentation and biotransformation. The third one is red biotechnology. It deals with medical and health sciences. And then gray biotechnology is environment and ecology, where we study about the pollution, the climate change, global warming, all those things we study on the gray biotechnology. And white biotechnology is industrial biotechnology, where we have processing and production of chemicals. Gold biotechnology is about bioinformatics, nanotechnology, where we study about the 3D structure of various biomolecules. Blue biotechnology pertains to aquaculture and marine aquaculture, where we study about marine and freshwater organisms, fishes, marine animals. Brown biotechnology pertains to arid zone or desert biotechnology, where we have crops, animal management in such a situation. Purple biotechnology is one where we study about publications and inventions. This is very important. All the students need to look into this every time. This is intellectual property right. So every student can really look into this purple biotechnology. You can patent certain things which you observe day in, day out in your life. This also is one good area where you can really work on. And then the dark biotechnology is what we now try to study, is about biological weapons, bioterrorism, which is not the right one. Even corona is also said to be a part of this biological weapon. We're not very sure, but whatever it may be, we see dark biotechnology is creating bioterrorism among the human population. So I have a list of 10 colors of biotechnology for human welfare. So I will be dealing with first 4 today. I'll be presenting various aspects of that. Okay, the first one is green biotechnology. Yeah, so this cotton plant, this is called boll. And this boll has a larva entering in and then eating all the seed material, the fiber, the cotton fiber. So this is a very dangerous pest on cotton. Net result is at the end of the harvest, you will not get a good quantity of this particular fiber, which is very important for us. So this is a picture that happens in every country where cotton is cultivated. But in order to overcome this situation, scientists have engineered a gene called crygene. Crygene is called crystal protein gene. Okay, they have isolated this gene from a bacteria called Bacillus thuringiensis. From this bacteria, they have isolated a gene called cry gene, and this cry gene produces this protein called crystal protein. And when the larva starts to eat the leaf, the plant starts to produce crystal proteins. As a result of this, the larva dies. Okay, so this is the whole story. Finally, the larva dies because of septicemia. That is how we are able to overcome a pest infection in case of cotton. And today we have DT cotton grown in every part of our country, especially in Andhra Pradesh and Telangana. You go to any village, you don't have the regular varieties of cotton, but you have DT cotton, and farmers are getting good yields in DT cotton. So far, there have been no reports of adverse situations on this, because this is not a edible crop. This is a crop for commerce, so there is no much damage done because of this genetic engineering method. Yeah, the second one that I would like to tell you is about brinjal. Brinjal also is infected by the same kind of pest. This is called fruit and shoot borer. So it bores into the shoot region, and this is how it lays eggs, and then the larva come out, and the larva starts to eat this. This is also the shoot. This is how the larva is eaten. This is a fruit borer, same borer. It also bores into the brinjal fruit here. This is another brinjal fruit. So all this is because of Licinoides orbonalis. This is the pest. So when you cut open it, you see the seeds and the pulp is being eaten away, and then you have the larva pupa inside it. So this is what happens with the brinjal. So scientists, this is the Licinoides orbonalis. This is the fly, the insect. It lays eggs. This is the larva, pupa, and then the adult. So this cycle goes on. So it can have 1 or 2 cycles on brinjal crop every time it infects. So this is one area. So what scientists did is they have developed BT brinjal. Why I have not given you BT brinjal details is BT brinjal has not been released in India. There is a moratorium on this particular BT brinjal because it is a vegetable. So there is a lot of discussion going on whether we should release this for the market, for the population. A lot of things are being discussed, so it is not released yet. But Bangladesh and other parts of the world are having BT brinjal. It is used as a vegetable. BT brinjal is used as a vegetable, and there is no side effects because of the genetic engineering method. The third one is what we see is this golden rice. This is very important. You see a kid here. Usually in Africa, thousands of kids die every year because of vitamin A deficiency. They have blindness. So Potricus from Switzerland. So see, Switzerland does not grow rice. Switzerland grows wheat. But see the interest of this scientist that wants to save people, children, especially from third world countries in Africa and India, where we have rice as a staple food. So they have engineered vitamin A gene into rice, and now rice is golden rice. I'll show you a picture of that. So again, this is also because of genetic engineering, and because of this, now African countries are growing golden rice. In India, golden rice has not been released yet so far. Okay, so the fourth thing I would like to suggest to present here is there is a lot of micronutrient deficiency. This is called hidden hunger. About 190 million children are suffering from the want of major micronutrients and vitamins. Okay, so vitamin A deficiency. This is because of vitamin A deficiency. See, as we have rice as a staple food in our country, especially South India, in Africa, banana is staple food. Yeah, sorry. This is the wild type of rice. And this is the golden rice one, where we can see faintly vitamin A, and this is golden rice 2. We can see intense golden color. This is because of vitamin A. So now this has not been released in India, but is written elsewhere, right? So this is what I'm trying to tell you is golden banana. What is golden banana is, see, in Africa, as we have rice in our country, they have banana in their country as staple food. Banana is also water intensive crop. Please understand this. As rice is a water intensive crop, banana also needs a lot of water. Okay, but this is a staple food. So what scientists did is they have introduced vitamin A, provitamin A, PVA, into banana. So when they have introduced into banana, banana started to express. See, this is how it started to express. So using transgenic technology, they have been able to develop this golden banana, and this golden banana is now enriched with vitamin A, and the population can consume now a banana. That is what is being done in case of some African countries. See here, this is transgenic banana and being explained about how transgenic bananas have been developed. Yeah, from here, what are the emerging trends in green biotechnology? One of the very important thing that I would like to suggest today is conservation of biodiversity. I have some pictures at the end, I'll show you them. Conservation of biodiversity. We need to protect flora and fauna from being endangered. Then we need to produce colored cotton. Why we need to produce colored cotton is artificial dyeing of cotton has created a lot of problems, health problems. They have caused cancer. So we don't want artificial dyeing. We want a natural blue colored cotton, natural red colored cotton, for which we are trying to use RDNA technology. Another very important area that we need to understand is degradation of plastics. We need to develop recombinant proteins in bacteria using genetic engineering methodology to dissolve polyhydroxybutyrate PHP. This has become a major pollutant in all the states of our country. It is found that plastics are being eaten by cows, by buffaloes, by cattle, by any kind of animal, which is now going to cause a lot of problem for us. So this is one area where we need to study. These are the trends, emerging trends that are happening in green biotechnology. Then we have avenue plantation. What is a genetically engineered avenue plantation is the plants will have green fluorescent protein. So when we are traveling on the road and your vehicle will put its light, you have this radium fluorescing. Now what scientists are planning is we'll have avenue plants with GFP gene, so that when lights fall on the plants that are on the either side of the road, they will fluoresce, they will highlight. So as a result, it becomes easy for the driver to see what are the problems or stumbling blocks there. Then bioinformatics is another area. Then biopesticide, biofertilizers, bioinsecticides. Then crops that withstand climate change. This is a recent one. Lot of climate change effects are being visible. So drought tolerant. Then we need to have hybrids. Hybrids are protoplast fusion. Hybrids are protoplast fusion that we need to develop. And then bioremediation. This is the use of vetiver roots to clean water, to purify water. So these are emerging trends in green biotechnology. So students can look into this kind of situation and also can undertake some projects. I'll be able to show you one or two at the end. Okay. The second one is I have gone to red biotechnology. Red biotechnology is pertaining to medical and health sciences. Okay. The first genetically engineered product is humulin. Humulin is the RDNA technology product wherein insulin gene is inserted into bacteria and bacteria starts to produce insulin that is called humulin. I'll show you a picture of that, how humulin is produced. And another very important thing is vaccines. Today we are very much interested in getting vaccines for corona. Not only for corona, we need vaccines for cholera, we need vaccines for different types of viruses, different types of bacterial diseases, TB. So we need a lot of vaccines. So can we use rDNA technology for developing vaccines is also being explored. Then gene therapy, wherein we introduce a gene, example in hemophilia, fibrinogen, I have also a picture of that. Then one very important area is your diagnostics, wherein polymerase chain reaction, which we call as RT-PCR now, is used for diagnosing viral diseases, also for DNA fingerprinting. DNA fingerprinting has come up, then protein footprinting, all these have come up because of rDNA technology. And finally, Human Genome Project. This is one area where we have to look at what is the Human Genome Project. A lot of information has been collected from this Human Genome Project, which went for 15 years. And the entire human genome, all the 23 chromosomes have been sequenced. We know where a gene is now, which chromosome, what is the size of the gene, what is the sequence of the gene. You have just to fish out from your bioinformatic tools, from public domain, and then we need to understand, we can use it for our research. So Red Biotechnologies here. This is, as I've told you, we are interested in DNA and RNA-based vaccines. So here you have DNA and RNA-based vaccines. This is whole vaccine, which you're not going to detail, but you have DNA-based vaccines and RNA-based vaccines. So we need to develop vaccines for different kind of viruses, okay? Because viruses cause a lot of disease on human. So people are trying to find out various methods of using this gene, how to develop vaccines. Yeah, Human Genome Project, I've told you that this is one chromosome. Today we know the chromosome DNA sequence. We know where is the location. This is centromere. This is P arm. This is Q arm. So we know what is the sequence. So all the 23 chromosomes have been sequenced. We know where is proto-oncogene now. We know where is the blue eye color gene now. We know what is the hair color gene now. Which chromosome? What is the sequence? It is now being available for research through Human Genome Project. Yeah, this is diabetes mellitus, a very common disease that is happening now with not only elders, but younger boys and girls also. So this is pancreas. The beta cells will release insulin, and then they maintain the level of carbohydrate or sugar in your blood sugar they maintain. You have diabetes 1, type 2 diabetes. So 2 types of diabetes are there. This is called diabetes mellitus. A lot of people are suffering. One major reason why people are suffering from diabetes is stress. Not just the other information, but it is stress. A lot of stress is there for people. So because of stress, they have become victims of diabetes mellitus. So this is the methodology. Recombinant DNA is introduced into bacteria. Now bacteria starts to produce insulin. This insulin is purified, and then it is called as humulin. This is called humulin. So humulin, human insulin, recombinant DNA origin. So this is how now it is available in the market. Earlier, we used to get insulin from animals. We used to get pancreas of animals and isolate insulin from that and use it exogenously, but it was causing allergy. So scientists have designed this. Eli Lilly company has developed this particular methodology, and today humulin is available in every medical store for treatment of diabetes. Okay, yeah, this is another one, another disease called hemophilia. What is hemophilia? Hemophilia is loss of blood on an injured part. Why loss of blood? Because there is no gene for producing fibrinogen. Fibrinogen is a clotting factor. So they don't have a clotting factor in them. So fibrinogen is not produced. Clotting factor has undergone mutation. So fibrinogen is not produced. When fibrinogen is not produced, then automatically what happens is the blood will go out. See, this is how you see. This is the bloodstream, normal clotting. But here there is no clotting, so blood will ooze out. This is the natural one that we have. So the kid is getting blood from his nose, from injured part, because there is no clotting factor. This exactly what happens is you have a clotting factor like this. So all the blood vessels, all the platelets, all the cells in the blood will be entangled here. But persons suffering from hemophilia will not have clotting factor. So as a result, what happens is the blood oozes out. This is become a very common disease. It is called royal disease because mostly it happens in royal families. But see, the kid is suffering from blood coming out from his nose. Yeah, so what scientists have done is, from Human Genome Project, they have isolated this gene, the gene for fibrinogen, and they have inserted that gene into the hemopoietic cells where blood is produced. So that is being worked out. We hope that in short, hemophilia also will be corrected through gene therapy. Yeah, and then this is called aging. A lot of people age from when they move from senior citizens, they become age. See in this picture, see this is a youthful face, while here you have an aging face, you have wrinkles, okay, and then you have jaws having foldings. This is happening, okay. So what scientists have found is telomere region in the chromosome, telomere region in the chromosome is responsible for aging. Why? Because at every round of replication, the genes that are present at the end of telomere region are lost. So because of this, they say that aging is introduced into human. As a result, they have developed a technology called TERT technology. Using TERT technology, now they want to reverse aging. They want to make people youthful, though have traveled for 60 years or 70 years, they want to bring back, and that is being worked out by reverse aging is done by TERT therapy. Again, this is genetic engineering method. Okay, I am showing you one very important aspect of yellow biotechnology. Yellow biotechnology does deal with fermentation, biotransformation, also wine making, but I'm not going to deal with that. I'm going to deal with this one particular aspect, that is the Indian honeybee. This Indian honeybee is the best honey pollinator. It is called Apis cerana indica. Because of Indian honeybee and other pollinators also, you have seed production. All crops get seeds because these are the pollinators. Okay, but their numbers are declining. The numbers of honeybees is declining. Why? Because of human anthropogenic activity. We are doing a lot of anthropogenic activity. We are using pesticides, so they are not able to tolerate, they die. And then European honeybees are attacking them. So this is what we need to understand, that we need to protect honeybees. How? I'll tell you in the next 1 or 2 slides, right? So we need to have some biotechnological tools to protect these honeybees. Okay, right. For those who have not seen, I am showing you Lupha cylindrica. Lupha cylindrica, male flower. Lupha cylindrica is Bira, Neti Bira in Telugu. Tirupati and Sujata, they have worked on this particular plant. Okay, I had the opportunity to look exactly what happens during the process of pollination. So I will show you this particular video. Okay, so this is the first video. Now see the second video. This is a female flower. Yeah, what we find in this is honeybee is intelligent. Why? Because male flower, honeybee knows that there is pollen grain as well as there is nectary glands at the base. In order to release the pollen grain from the male flower, it will vibrate with its wings. That is what we find in case of first case. Why this is vibrating? Why is it vibrating? Is to see that the pollen held in this pollen grain in this anther will be released and they fall here, so that then it can collect. How intelligent is this particular insect pollinator has to be very much appreciated. In this case, you don't find that it will hover over it. It just comes, picks up the nectar, and then leaves. That's all. So that is what happens. So these are very intelligent beings, very intelligent beings. So how do we protect as students? This question was asked in the last meeting also. Students can visit your sunflower or some crop in the nearby fields, and they can go for apiculture. Thereby you're protecting these insects. Very interesting and useful insects, that is pollinators. Indian honeybee. If you want to do some small projects, you can have a small apiculture unit in a nearby place where you have a small farm. Then you can also study how intelligent this particular insect will be able to perform. Yeah, and the last one is the gray biotechnology. Very distressing, very damaging. What is that? Is lot of oil is spilled into the sea. These are the ships. The ships collide. As a result, the oil is spilled into the sea. This is the net result. Birds die. See, this man is trying to protect, trying to remove, but this is disastrous to marine life. See, this is a tortoise. Okay, tortoise is covered with oil. Some persons are trying to see whether we can remove oil. The animal, the tortoise dies. Bird has died. This is a cormorant. It has died because of this particular. And then surf scooter also has died. He is going to die because of this oil spill. Okay. In Gulf of Mexico, they are trying to spray some oil decontamination oil that is called Corexit, but it has not solved the problem. So in this aspect, you should remember Anand Chakravarti. He created Pseudomonas putida, or he engineered Pseudomonas putida. What is Pseudomonas putida? It will gobble up oil from the ocean. His is the first patent awarded for a living organism in the whole world. Anand Chakravarti was granted a patent for engineering Pseudomonas putida, which will gobble up, which will remove oil from the ocean. Okay, so this is the technology that he has used. Finally, he found that he can degrade camphor, degrade octane, xylol, naphthalene. So he got strain G, and now this can be used to remove oil from the sea, the ocean. This is being explored, and I think in course of time be useful. Okay, right. Yeah, I've come to the end of this small lecture. What I'm showing you is a museum. A lot of birds, animals. This is not in our country. This is elsewhere. Most of these are endangered. Most of these pictures that I'm showing you are endangered. Why? How? I have no answer for that, but they become endangered. One very important thing that I would like to tell you is about our house sparrow. Okay, so house sparrow, these are having a good time. These are the male and the female. The mother is feeding the chick. House sparrow are affected because of antennas, where we have your vibration, where we have radiation. Okay, so if this is a situation, in due course, what they're saying is 4G technology might be affecting them. Now we have 7G technology. What will happen? All these birds will be exterminated sometime. So what as students that we can do is we should feed them, give them some water. If you come across group of birds there, you can give water, you can give them some grain. So thereby these birds will be protected. Here a person has given a home for these birds to stay. I'm giving you one example which I from my childhood have seen a lot of these house sparrows, but today in my locality, in my place, I don't find them. Maybe you can find them in the fringes of the cities or towns, but where you have villages, but I don't find them now in my place also. So as students, we can protect them. As students, we can help them by providing grain and providing water. This is one area where you can do a small project for your MSc, for your BSc. Here, this is a place where you see in Chennai, all the students have formed a awareness program where they formed into a bird, that is the safe bird concept. So this is how they are trying to educate people to protect birds. Okay. Another very important thing is, this is a common sight in everybody's house. We have your tulasi tree, and these are plants. Every student has to have this kind of growth. We need to protect plants. We need to grow plants in our backyard. We need to have all kinds of plants, ornamental plants, fruit plants. See how beautiful this particular picture gives us. So at least from the point of view of our religious sanctity, let us protect our plants. These are very, very indispensable for future generations. We lose them, I think we are going to, doomsday is not far away from us. Here, this group of people are donating trees, smaller saplings, medicinal plants, free donation. They are not charging. This kind of attitude has to develop for our students, so that we need to protect. Mana intlo unna vaatni kuda protect chesukochu. We need to do this. Also, we can give some plants, tulasi plants ivochu, lekunte inka bere mokkale emana unte ivochu, medicinal plants unte ivochu, aloe vera ivochu, xerophytes ivochu. So we need to understand that grow medicinal plants and distribute free to people. At least by doing that, we'll be able to protect them in our kitchen garden, in our backyard. Yeah, so the last slide is all for one and one for all. Keep biodiversity or our future may fall. Okay, so some future rDNA technologies, they're going to say that we have introduced a green fluorescent protein gene into mice, and see the ears and the eyes of these mice are fluorescent. Okay, so they can be used as models. Yeah, you're going to create transgenic birds. See here, they have introduced a green GFP gene, green fluorescent protein gene. So the chick that is produced here is green in color, green fluorescent chick. And several approaches are being used for developing a vaccine for viruses. So the end is namaskar. Thank you very much for your patient hearing. Any questions, I'll be able to answer.
Thank you very much, sir, for your enlightened lecture.
Yes.
Participants from the student side, any queries? You can please ask sir, got any questions you want to raise the questions. Students, if you have any doubts in this talk delivered by sir. Sir, there is a question from Akhila Sharma, one of the students.
Yeah.
She's asking, how does the fluorescent genes
Yeah. This will help us for making corrections in some diseases. See, normally we develop models, mouse model, chick model. Models are very essential. Without models, you cannot do it on human. So these are models. So what scientists are trying to see is, can we introduce green fluorescent protein gene into a chick and see that it expresses in a chick. Now then they will transfer this onto human. So suppose we are trying to transfer fibrinogen gene. How do you know that fibrinogen gene is there or not? Is it should be tagged with green fluorescent protein. So that is how it is useful as a model. We are not going to produce a fluorescent chick. We are not going to produce fluorescent chick. But in fishes, they are producing green fluorescent fishes, because ornamental fishes are there. They're growing in the aquarium. Ornamental fishes are being. So this acts like a model for further experimentation.
Thank you, sir. Students, still any more questions? You can chat in the box, put the message so that I can read the question and answer the question.
Sir, thank you, sir. This is Dr. Achita, Principal, R.B. Vera Women's College, sir.
Madam, thank you so much. Thank you so much, madam.
Thank you, sir. Thank you very much.
Thank you, Principal Ma'am, for attending our webinar.
Yes, madam. Yeah, one take-home message to all the students is, we need to protect our birds. We need to protect our plants. We need to protect our environment. It is in your hands. By giving water and grain to birds, whatever is a type of bird, it is going to be useful for the ecosystem. So students should take up small projects on those. They can do apiculture, then go to a field and see, maybe in future, ants are also useful in protecting plants. I have that in the next class, maybe future. Ants, మనం అనుకునే చీమలు వీటి వల్ల ఏం ఉపయోగం లేదని అనుకుంటున్నాం. But even ants also have a lot of things to do for protecting crops and plants. I'll tell you in future if we have such a situation also.
Okay, thank you very much, sir, for your nice conversation and the message delivered to our students of conserving our nature.
Yes, madam. Yes. Thank you.
Now, I request Dr. Tirupati, sir, to unmute yourself and propose formal vote of thanks. Over to you, sir.
Okay. Good evening, Kavita Madam Garu. Very good evening to all. Respected today's chief guest and keynote speaker, my godfather, Professor E. Christopher Reuben, sir, has delivered an excellent lecture and very hot topic and emerging topic, that is emerging technologies in green, yellow and red biotechnology. This is very, very useful for all the students who are interested to becoming a researcher, becoming a scientist and research fellows and teaching faculty. And even though the all science students is very, very useful for today's lecture. And sir has explained one of the important area, that is how to conserve even though the honeybees and conserve the house sparrows and ants also. So these are very, very important for future generations for food security. So without these very important pollinators, without these honeybees, even though house sparrows, so we cannot develop or we cannot survive in the future because of so the insecurity of the food. So we are very, very suffering, lots of facing lots of problems. So that's why so today's lecture is very, very useful for the conservation of the nature and also how to conserve the biotechnologies, the new emerging technology, that is green, yellow and red biotechnologies. These technologies is used for conserve the nature and conserve the all living organisms for future generations. So sir has also discussed in my PhD program. Sir is also explain how to conserve even though the honeybees, sparrows and the entire nature. So sir is very, very kind-hearted and thinking about the nature and also the downtrodden people. So sir has given an extraordinary lecture and we are very thankful to sir because of sir today's suffering severe cold and fever, but sir has committed sir his commitments and dedication towards the what sir has given to word to given lecture. Sir has given this 1.5 hour without any interruption. So because of we are very, very lucky and we are very thankful to our Professor T. Christopher Reuben sir. So his commitment, his dedication, his determination, his role model and inspiring us as a faculty of higher education department. So we are never forgettable your words sir. So we are very lucky and I am also one of the research scholar of my professor, my godfather. I am very lucky. Sir, sir has giving us valuable time for us. So thank you very much, sir, for giving this lecture and your valuable time for us. Thank you very much, sir.
Thank you, Tirupati. Thank you, sir.
And we are very thankful to our today's pattern, Dr. J. Achyuta Devi Madam Garu, principal, one of the prestigious institution in Hyderabad, that is RBVR Women's College, Hyderabad Autonomous, and principal of Government Degree College for Women, Jagityal, Dr. Yamsani Satyanarayana Sir Garu, giving me for conducting this one-day national webinar on emerging technologies in green, yellow, red biotechnologies. So we are very happy and thankful to all the organizers of today's webinar, that is convener of today's webinar, Dr. Dr. A. Rajini Madam Garu, Head Department of Botany and Food and Nutrition, RBVR Women's College, and co-conveners of today's webinar, Dr. A. Kavita Madam Garu, Department of Botany, RBVR Women's College, Dr. Manjula Madam Garu, RBVR Women's College, Hyderabad, and Vani Madam Garu, and other lecturers, other faculty of Department of Botany, and also my department, Dr. G. Chandraya Sar Garu, Head Department of Botany, Women's College, Jagityal, and my senior faculty of Zoology Department, Dr. A. Jyotilakshmi Madam Garu, Dr. K. Kiranmay Madam Garu, Life Sciences. So we are very happy and thankful to each and every teaching and non-teaching faculty of both the colleges. And also my sincere thanks to Professor Sudarshan Reddy Sir Garu for his kind-hearted and words regarding of making these two institutions in collaboration manner and conducting such a wonderful programs, even though PG get coaching and other competitive examination coaching also. So each and every faculty and also participants of today's webinar and students, we are very wholeheartedly thankful to you, each and everyone. Thanks a lot. Over to Kavita Madam Garu.
Thank you very much, sir, for your formal vote of thanks to each and everybody. I personally thank our secretary cum correspondent, Professor Sudarshan Reddy sir, and Principal R.B. Vera Women's College, Dr. Achutha Devi ma'am, and Head Department, Dr. A. Rajni ma'am, for giving us this opportunity to organize this webinar in a smooth manner. And I personally thank Professor Christopher sir, in spite of his illness today. He has delivered an excellent lecture, and I hope all my students have taken up your valuable words and the knowledge from your sir. Sir, please, sorry, we make you to do this webinar.
No, madam, no, madam. You're welcome. You're always welcome.
Get well soon, sir, and so that we can have more interactions like this in the future. Thank you very much, sir, once again.
Thank you, madam. Thank you so much.
Yeah.
Thank you, sir. Thank you so much, sir.
Madam, thank you. Welcome, madam, and Rajini, madam, thank you. See you again.
Sir, one screenshot for this one, sir. Sir, Tirupati, sir, you can also switch on your video so that we can have a screenshot.
Dr. Parvati, Dr. Malati are also there today.
Vani ma'am. Vani ma'am also. Vani ma'am also. Ma'am, please you can switch on your video for the screenshots. Done? Done? Yeah. So thank you very much, sir.
Okay, madam. Thank you so much. See you again.
Sir, thank
Yes,
you so
sir.
much.
Sir, thank you, sir. Thank you, sir.
Thank you, ma'am. Thank you.
Tirupati, sir, thank you so much for organizing.
Thank you, madam. Thank you.
Nanda ma'am and all the other faculty members of R.B. Vera Women's College who have joined this webinar, and also from Government Degree College and other ma'ams from the other departments. Thank you for joining this webinar. Thank you very much, ma'ams. Yeah. Yeah, the session has been ended. Naz, you can stop recording.
Okay, ma'am, stop it.
Yeah.